So we'll let each person in turn give a brief intro and who they are and why they are in IO. Some are more obvious than others. And then we'll start with the questions. Please, at any point when I'm having questions directed to the panel, feel free to jump in with your own follow-up questions or any additional questions. Just raise your hand and I'll try and see you. So, Paul.
Thanks, Jeffrey. Paul Biondi, I'm head of our business development group for Bristol-Myers Squibb, and that includes really all of our business development activities worldwide, spanning from the search and evaluation of new opportunities to either licensing or partnerships or acquisitions through alliance management. I've been with Bristol since 2002. I was actually on the R&D side in kind of strategy and portfolio management operations for many years and took over business development last year.
Thanks, Paul. Niels Emmerich from AbbVie in Chicago. I'm heading search and evaluation for oncology at AbbVie, so focused exclusively on oncology. And we support all types of transactions from discovery collaborations all the way to mergers and acquisitions, as long as it's oncology, which is a therapeutic area of growing importance to AbbVie next to immunology and neurosciences.
I'm Stefanos Thecharis. I'm Senior VP for Corporate Development with Cell Medica, responsible for in-licensing and out-licensing. We recently did a large umbrella collaboration with Baylor College of Medicine for CAR-T and CAR-NKT cell development, another collaboration with UCL in London for TCR-modified T cells, and still looking for a lot of technologies to bring into these T cell technologies to increase specificity, to decrease the side effects, and also combination treatments with some of our non-modified cells, for example, checkpoint inhibitors, where we see that a T cell product would increase the longevity of response for a lot of the patients receiving checkpoint inhibitors, and any other relevant technologies. I'm here scouting, finding out what's going on in the space.
Good afternoon. My name is Bernd Mühlenweg from Nanobiotix. Thank you very much for joining the show, first of all. Nanobiotix is developing a compound which is made for enhancing the effects of radiotherapy. This is an enhancer where radiotherapy cannot bring enough efficacy or where radiotherapy is too toxic, so that you try to reduce the dose of radiotherapy, while adding our compound brings it to the same level of efficacy. And this product is now in registration in Europe. And roughly a year ago, the big pharma companies started to combine radiotherapy with their immuno-oncology programs. And today we have 90 programs utilizing IO plus radiotherapy in clinical development. So this is the reason why I'm representing us here, because we have the product that can even enhance further the radiotherapeutic effect, and there with the immunogenic cell death, which opens new windows for immuno-oncology compounds to be combined.
Thank you, everyone. So just a quick show of hands here. How many companies or people in the audience are directly or indirectly involved with immuno-oncology? You have an asset, you're a biotech, okay. How many are absolutely opposed and don't believe that immuno-oncology works? All right, no contrarians. All right, well, so as I said, I think it's valuable that we have both biotech representatives to kind of showcase kind of their view and perceptions of what it's like being a small player trying to get large pharma interest in their technologies or platforms. And we have two very large oncology players with investment, not just in IO, certainly, but beyond that, because one of the things we want to get out of this discussion is what is of interest to them when they're looking outside, since we have search and evaluation represented here, in terms of those companies that have small biotechs. And so I think we'll start there, segueing from what Bernd said about their approach and the idea of combinations, because it really does now all start with combinations. Not to say there's not value to proving your single agent monotherapy activity, and there might not be applications of that going forward, but by and large, we're looking at what will likely be an evolving combination therapy world, whether it's checkpoint with checkpoint, checkpoint with co-stim, checkpoint with vaccine, et cetera. So in order to frame some of the more partnering related questions, I just want to get the viewpoint from each of our different panelists, from their different perspectives, how they're thinking and viewing this world of combinations, because we were talking about trying to expand the realm of tumors of patients who are responding to the agents we have, expand the reach of IO into the less immunoresponsive or so-called cold tumors. So let's start there to frame some of the scientific clinical aspects, and then we'll move into some of the BD-related aspects. So with that.
Sure, I can get started with our approach. Obviously, combination approaches from Bristol's perspective has been kind of the core of our strategy from the beginning. And one of the reasons that we originally did both the partnership and ultimately the acquisition of Medarex was because of their view in looking at these co-stimulation opportunities and how that could obviously stimulate the immune system to fight cancer. So I think we've learned a lot about, one, how to potentially approach doing combination therapy. I think in particular, have really focused on the power of the dual regimen of both the combination of nivolumab and ipilimumab, one of the things it took us some time to figure out is actually how ultimately to both sequence and to dose it. I think we're really pleased, particularly what we've seen in early data in non-small lung cancer with the combination, but also it's bearing out in multiple tumor types now. And I think that kind of development approach is really important because we're seeing differences as you move across, potentially, different tumor types and what's the right way to combine. One of the things we've tried to do to accelerate, because there's so many opportunities, and we have a large portfolio, and we're interested in partnering with many of you to find these, all these different unmet needs, because even though we've made some interesting gains, obviously, in these kind of durable responses, there's still a huge amount of unmet need when you look across tumors and even in those tumors that have had a response. So, part of what we have done is try to come up with an innovative clinical trial design. There's one that we call Fraction, in which really pretty rapidly allows us in an early clinical environment to evaluate how do you think about the current standard of cares from our portfolio to then to say, hey, where could we advance, shut down arms, and move to others in sort of an adaptive-like approach. So I think that kind of not only having the preclinical piece, which gives you some biological plausibility as to, perhaps where we should go, but I think we all recognize we're still learning a lot around the biology and certainly around the nature of the immune system and pre-existing status of it versus, tumors. But how to then take that and advance it rapidly so that you can do some of this clinical investigation. So that's a lot of been our approach.
Yeah, I think at AbbVie we fully agree that combinations are going to be key. And if you look at how the immune system is wired, there's just way too many negative feedback loops that start to kick in once you trigger one particular target and pathway of interest. So to combine in a rational way with synergistic activity as the ultimate end goal, different targets and pathways and drugs against those appears just from our understanding of the science the way to go. And we really do that and I hope you'll hear a lot more about what AbbVie is doing in the cancer immunotherapy space in the not too distant future. We're really approaching that in two different ways. One is investments into first-in-class assets, novel targets, compelling biology and science on the one side, platforms that could lead to completely novel asset classes that we believe have a strong rationale for being pursued, but then also combinations where we see a strong rationale, such as the recently announced combinations with nivolumab of some of our later stage clinical assets in oncology, like inhibitors of DNA damage repair, PARP inhibitors, veliparib in particular, as well as drug candidates that we believe hold strong potential to induce in situ vaccination and lead to significant release of antigens locally to prime the immune system, especially in the presence of immunomodulatory drugs like nivolumab. Rova-T, the asset we acquired through our acquisition of Stemcentrx, is one of these examples.
So great. Then just make a quick comment before you take this to Stefanos and Baron as well. So I think from the small biotech company side, it'd be very interesting to hear, and I'm sure the audience would be interested, how you're thinking about these combinations from your perspective, because clearly, from a well-resourced large pharma company, there are many things they can do. From a smaller company, your choices have to be well-made, and there are a variety of ways you can access those potential combination partners, whether you have them internally, access them through clinical collaborations with a large player, other opportunities. So if you can address that, Stefanos and Baron, in your responses.
Yes, and I mean, I would actually consider us to be a well-resourced small company, and we actually are quite well-financed. And I think, you know, a company in our stage and in our technology is reasonably well-placed to raise capital to develop a lot of these therapeutics. So a lot of what we do and the combination isn't purely a financial combination. I think, and if you see our pipeline, everything we've done to date is combination with experts in different areas where they can actually contribute a lot more than just financing. And so in the combination space, which is not the whole story for us, because obviously with the CAR-Ts and TCR-Ts, they are, you could call them a combination product itself. They have so many layers of technologies in them. But looking at technologies, for example, such as checkpoint inhibitors, it's something that I wouldn't say stumbled upon, but working with experts at Baylor College of Medicine, with whom we're working on our EBV-specific T cells, they came back and said, guys, these tumors are very highly expressing PD-L1. You guys really need to consider a combination with a checkpoint inhibitor to actually, on the one hand, release your therapeutic product, your EBV-specific T cells within the tumor and allow them to function. Whereas the theory, on the other hand, the corollary to this is that by generating the immunological memory that the T cell would be expected to generate within a patient, then you have a greater longevity of response with the therapeutic checkpoint inhibitor. So it kind of makes sense on paper. We have some data to show the scientific rationale. Now for me it's interesting to speak with people who are experts in the checkpoint inhibitor space and understand the targets, the patient population, the patient subpopulations, what are the challenges in a combination treatment, so that we can actually design a much better therapeutic together. That is really the knowledge that we will be gaining with the partnership is much more valuable to us.
Thank you. And of course, for those maybe not as up on adaptive cell therapy approaches, aside from the ability that Stefano said to do a combination, say, with an extant player partner like a Nevo, et cetera, the technology is moving so quickly, some companies are engineering in those checkpoint inhibitors right into their cell therapy constructs.
A lot are, and it's obviously, and well, that's obviously a way to ensure that your checkpoint inhibitor is released at the right place, at least where it's needed in the cell that needs it at the right time. And obviously, it spills over to other cells in the immune system, which would be infiltrating the tumor. Another thing that companies is doing is they're gene editing the receptors for these checkpoint inhibitors out of the T cell, and that will obviously benefit the T cell itself. But that effect will not spill over to the rest of the immune system, who would also benefit from a checkpoint inhibitor. And then you could also consider a combination or the additional engineering of a single-chain antibody against PD-L1 or PD-L1 being secreted in the tumor. There's a lot of options that we haven't looked at, but obviously all of us are going to be thinking about all of those options.
Yeah, and now, I mean, very interesting, particularly for the audience, to hear what Bernd has to say, and I think Niels kind of opened up the potential here about the idea of combining IO and non-IO agents. So that includes, as Bernd will talk about, radiotherapy, but obviously things that can induce or de-repress DNA replication and increase the mutational load, and et cetera. So.
Yeah, thank you very much for this introduction. I mean, yes, we are also strong believers in the combination, but not in this combination like you described it, where you essentially try to tackle several targets simultaneously. Now, independent of the background technology, let it be an antibody therapy, whatever it is. What we are trying to do is we are trying to open another perspective of combination where we try to prime the immune system with enabling technology, in our particular case with radiotherapy, with a simple idea to make to get a larger patient population eligible, or even in the scope of immunomodulatory agents, because we know that only the minority of patients today are really eligible for these kind of approaches, and many still don't respond. So with our approach, we are trying to use radiotherapy, an enabling technology, to prime the immune system to stimulate the immunogenic cell death. And once the immunogenic cell death is stimulated, then we would add, let me say it a little bit negatively, the classical approaches that you are developing on top of these stimulated immune system. Now, in our particular case, the problem with these approaches generally is that if you use radiotherapy in combination with any kind of PD-1 inhibitor or whatsoever, you cannot really patent this. And this is essentially what we try to do by utilizing our compounds in combination with radiotherapy to open a new combination approach, which is patentable in combination with immunological agents, so that really it's an increase of the patient population. It's to provide more efficacy to the patient, more benefit to the patient, and at the same time addressing the uncertainty that competitors cannot really go into this pocket and differentiate through a patented approach here.
Any questions at this point from the audience? Or? So I think you, Paul, mentioned the fraction approach and the idea of these adaptive trials. Certainly in both a positive and a negative way, people have commented about some of the shots on goal or the necessary empiricism that's happening right now in immuno-oncology, because you can only take the models so far. So there's a lot that has to happen at the clinical stages to really see if there's any traction with, particularly with novel targets and in combination. But that being said, you know, any small biotech out there is still stuck in a situation where they're having to be dependent on early model data, whether that's xenograft or syngeneic, et cetera. So I'm just curious to get kind of the two sides of the story from the large pharma players about how they kind of view those early signal generation in model systems and what they're looking for, and similarly kind of the flip side from the smaller biotech, you know, how you think about, you know, doing that preclinical work and what you feel you need to do and what you've been hearing from prospective partners about the pros and cons of how you've had to do those preclinical models. So, Paul?
Yeah, I mean, I think that, you know, definitely there's going to have to be, and we continue, as I think we continue to understand exactly how the immune system works, and as we get, as the nice thing about success in an area, it sort of highlights, okay, the areas that it's not working, you know, what are the things that might be driving that, and how do you get underneath it? I think that from our experience, which is, you know, to the extent that there's a lot of like-mindedness between, I think, our folks internally, preclinically, already thinking about some of these hypotheses, and then meeting with somebody externally, and then bringing data that kind of reconfirms that and the rigor that we would see it. Oftentimes, you know, that can actually work out well. You know, I think where there's something that is perhaps plausible, but yet not something that, you know, we've said to ourselves, hey, that makes a lot of sense out of the gate, you know, to try that approach. That's where I think it's helpful for us to say, you know, hey, generate some early clinical data, give us, you know, some reason to believe that, you know, this could have, you know, some impact and some synergy, perhaps with what we could bring to the table as well. So that's often a lot of the conversations that we have. Again, I think that's not to say, hey, don't come with us if you've got interesting preclinical, you know, data that might be interesting, but oftentimes you might say, hey, might benefit you to go back and generate some of that clinical data. One of the things we are trying to do is be, I think, more open and helpful in providing things like nivolumab, or perhaps nivolumab plus ipilimumab to folks, and, you know, just so that you could run, you know, drop that into your, you know, phase 1 study and generate some of that data in a more rapid way. So that's one of the things that we're trying to do to enable some of this experimentation, recognizing for limited resources in small companies.
Yeah, I think we all recognize that the old wisdom that if I was a mouse, I would be cured from cancer is still true, right? So there is inherent bias and to some extent lack of predictive value in doing mouse experiments, especially for immuno-oncology drugs. But to the extent that you can use syngeneic models, xenograft models, PDX models, we believe that potential partners, as well as our R&D groups internally, should make dedicated efforts to getting us the answers that can be gotten with reasonable efforts from existing systems. Once you get to the point that you have to engineer more than 2 variables, you get into highly artificial settings, right? So in some instances, and cell-based therapies may be one of these examples, it may be warranted to skip some of these steps and try to get answers in early clinical studies. But overall, I think we're looking for the same level of scientific rigor, data without glaring gaps that we would be looking for with every other drug candidate and mechanism that is non-immuno-oncology. I think there's too many agents overall that get branded as immuno-oncology. I don't want to take the raise of hands by almost everyone as such, but we've seen spectrum-specific kinase inhibitors being relabeled as immuno-oncology, right? Yeah.
And again, I'll say what I said before. It really does depend on us learning from the experts in what we can do. So obviously, we're starting to think about checkpoint inhibitor combinations. What are we going to do? We're going to see how everyone else has been developing their own checkpoint inhibitor. We're going to call up a lot of the experts in the field. Obviously, our partners will already have a lot of understanding in this, and try to understand what we can do and what generates some value. So what we can do, we will absolutely do. But what's important is to try and prove this in patients reasonably quickly. So having a quick look in some patient populations where you would expect to have some therapeutic efficacy, potentially with, if there is a companion diagnostic, for example, that will show, these are the EBV positive patients, these are the high PD-L1 expressors, and we will be able to diagnose this and move forward quickly on a small trial and trying to understand what's going on. A lot of it is therefore going to depend on discussions that we're having with everyone who knows enough about the space and has patients that they understand and know how to treat. I don't think there's any fixed rules, and certainly with the T-cell therapeutics, there's very few models that one can use to generate sort of predictive understanding of what will happen. The understanding is, of course, if a T-cell meets its target, it should activate and kill the target. And so you start to understand from there why it sometimes happens and sometimes it doesn't happen. To a large extent, we have a good understanding of that, and we're building on that.
Yeah, so I think immuno-oncology has matured significantly over time, and so I think that the partnering thresholds from you guys obviously have been raised significantly, which certainly is not a bad thing. I think my experience is that we get a lot of positive support from departments like you're running yourself in your two companies, also from others, trying to give us guidance as to what they would like to see when you're really opening the dialogue. And I mean, it's really an open discussion, so that the wish list is, we have a long wish list from externals available from the pharma companies, and certainly we are not able to fulfill this wish list for staffing reasons, for financial reasons. And I think it's not so complicated to work on preclinical models or to invest some money here and there. Really for us, the threshold is as soon as we have to do a phase 1 alone, even if we get one of these expensive drugs for free, I think we have to ask ourselves the question, where is the 3 million euros better invested? Is it better invested in this study, which may be particularly driven towards one pharma company with an uncertainty of they will take it or they will not take it, or do we invest it into one of our own clinical programs that brings us closer to the market? And I think the support definitely is there. We are closely listening to this, and it's really highly appreciated. It's even sometimes educational, I have to say. And the views are very different in the different pharma companies we are talking to, so it's really surprising to see that there is not the homogeneous opinion on immuno-oncology. It's really diverse, and I think everybody needs to talk to the specialist in the pharma companies to get a picture of what is each individual pharma company looking for to make a decision where to go.
So I don't remember which of you said it, but certainly I guess to a certain degree, everyone kind of alluded to this, the ability to access some of the approved agents, since they are presumed to be foundational. Maybe we'll talk about that in a little bit later. So that in of itself maybe dictates some of the type of deal making that one has been seeing within immuno-oncology. You see everything from outright acquisition, often at a very early stage, to get access to a novel modality or target, like IDO agents, for example. And you see also a lot of more clinical collaborations that, at least publicly, may or may not have some type of options rights at the end. So maybe everyone can kind of talk a little bit about, well, certainly from the large pharma perspective, how you're determining, at least from your preference, kind of where you like to go between those outright licensing, M&A opportunities versus the more clinical collaborations as a function of kind of the risk of that target, as a function of the stage, which obviously is related to the risk. Are there other elements that go into that?
Yeah, I think that's a great question. You know, a couple ways that we think about it. You know, a lot of it is fundamentally based on kind of, you know, and I like Bernstein, which is, you know, this is still a pretty pretty wide open field, honestly. There's still a lot going on. There's still a lot to be learned, and I think this is why, actually, having the ecosystem of small companies doing very innovative things, and probably I would say almost different businesses from our core business, from what both Stefanos and Bernd do, is a very healthy thing. So partly it starts from us with, I think, a conviction about sort of, how do we see the science internally? And then often we're reaching out kind of proactively to folks to say, hey, we're really interested in this. What's your view about partnering? What's your openness to maybe something different? And oftentimes that will be the genesis of an acquisition, because we just feel like, hey, if we could take your capability, your insights, bring that in-house, we think we can actually speed this much faster to patients in that approach. Now, that doesn't always fit in every case. Then I think we would also obviously look for, where can we do, I think, innovative partnering with where we have shared economics, and we're going to think to take this all the way together. I see Tarek there from Brian. There's a good example of how we've done that. And then I think what's nice about clinical collaborations is that allows us to do a whole series of different types of frameworks. Niels and I can do, we've actually done two together very rapidly to put together, hey, let's, you're going to have your assets, I'm going to have ours. It would be very compelling to look at the combination of Nivo with a PARP and, and what that could generate. That makes a lot of sense to us, which is, hey, we could generate that data and then figure out a collaboration, beyond that. It could be that, hey, that's a very interesting approach. We did a deal with Cyoxis, which they have an oncolytic virus approach, which we have some interest in around inflaming cold tumors, and wanted to see that data generated. So I think that's, gives us a way to essentially de-risk on both sides, to see, hey, as Stefan was saying, hey, does adding a checkpoint in or adding something else from our portfolio make sense? And then we could think about how to make this work. There's always, it's easier, obviously, always to have control and everything over, a portfolio, particularly as you're unsure as how things are going to evolve by tumor type. But at the same time, I think we recognize that in this world, that's probably not going to be the way that it plays out in every instance. So I think we can learn a lot actually from the HIV experience, where we and lots of companies, we all partnered to basically come up with much better regimens over time. We partnered with Gilead, one of our fiercest competitors, to make a triplet an incredible success. So, I think it clearly can be done, and that's probably what it'll take in the future.
Yeah, and I think it's becoming, it was not so common in the old days, certainly in oncology, to do those large pharma partnerships, notwithstanding, some of the Merck, AZ deals, some of those, but certainly the climate for those combinations from a regulatory standpoint, from a philosophical standpoint, from a necessity standpoint, I think, particularly driven by the realities of IO, is making those type of large player interactions and partnerships, I think, more likely now, which I think is a good thing. Anything else?
Yeah, totally agree. I think there's one risk here, and there's published data on that for oncology that tells you that if you combine with standard of care, then you have a 50% chance that standard of care will change by the time that you get your combination approved, right? Which is an additional risk in addition to the regular technical risks that you face in development. So we really hope that some foundational assets like the ones BMS and Merck brought to the market are here to stay, because we build in some of our clinical trials on those combinations.
Agreed on that.
And I think you mentioned Cyoxis. I think you're also combining with, is it Prostvac?
Yes.
So another virus-based product candidate. I think we see a resurgence of those, and we'll see more of these in combinations. Approaches, strategies that haven't necessarily panned out in the past. Vaccines, oncolytic viruses, danger signals like TLR agonists. But if you look at Novartis and Aduro, or the virus combinations BMS is endorsing, then I think this is one of the interesting and logical expansions of which we're going to see more in immuno-oncology combination studies.
Yeah, I'm glad you mentioned that, because it does emphasize, again, the role of different, first of all, it's not monolithic IO. We may focus on checkpoints, but it is a very diverse set of categories: oncolytic viruses, adoptive cell therapies, clearly, there's toll agents, there's even vaccines are widely diverse, even adoptive cell therapies are widely diverse. And I think that's a great advantage about the space as well, as you have this diversity. That's also a challenge, and I think we'll talk about that a little later for a large pharma player, particularly for new modalities. You know, how do they make a decision when to play something evolving very rapidly like adoptive cell therapy? If you haven't already made a bet, how and why, what triggers that? If you're in an area of checkpoint inhibitors, you know, you're asking the question, do I get in with the 6th or 8th or 12th agent and why? And there are pros and cons to both of those questions. But so now from Stefanos, I'm curious to hear your kind of comments on.
Well, in the point of when to jump, when to make a decision, I mean, we have to make this decision, you know, all the time when we look at the different technologies that we could or we should or we may not integrate into our T-cell products. And you know, the CAR-T and TCR-T space as it is at the moment, it might to some look like an arms race, right? And everybody's just accumulating different technologies. And some of them make a lot of sense, of course. And obviously with a lot of the CD19s out there, which by the way, I don't see the CD19 of a competition. It's a tiny disease target that everyone is vying for. It's a proving ground. This is where everyone is testing their new technologies on a validated target, and that's why there are so many of those. I mean, Juno has 4. And obviously they're testing, there's humanized and the human and an armored CAR, which secretes IL-12. So this is really the decision that we have to make. What technology do we need to add in order to either improve the specificity or reduce the potential risks? I mean, we bought a company in Switzerland. It was Delenex Therapeutics. It's a single-chain antibody-specific company. They have their own platform. And the reason we did that is because we believe that this is a technology that's absolutely crucial to the optimization of the CAR. So it's been shown by multiple groups that on the one hand, reducing the affinity and then optimizing the affinity of the scFv on the CAR will significantly increase its specificity. And that is absolutely necessary if one wants to move into the solid tumors, where you will have a target that is expressed, albeit at lower levels in normal tissues, and then a tumor where you have high levels of expression of that target. And in order to be able to understand how to do this and what are the parameters and what methodologies we have to establish in place, we felt that it's best to build that in-house rather than just go to a CRO and order some scFvs and then try and do the work in-house. We buy an expert company on scFvs and then we teach them how to test on CAR-T. So they're, in fact, I mean, they're all immunologists, so they really know what they're doing, but you know, we're bringing the cell assays there in order to test that. And you know, other things like what people are talking about, like tonic signaling, for example, the fact that scFvs will associate outside the cell, bringing a lot of the CAR molecules together, and that tends to signal and semi-activate the T cell, which by the time it reaches the tumor, it's exhausted and it doesn't work anymore. And you know, how do you correlate the measurements of a tonic signaling in a T cell with the biophysical properties of the scFv? You study the biophysical properties of the scFv, you put it on a CAR, you put it on cells, you test it in biophysical assays, and you learn that. So that's really, you know, that's where we made the decision. So if we could stack up all the sort of downstream milestone and royalty obligations from licensing an scFv versus buying a company and doing it in-house, the economics kind of made sense, but the real value for us was actually generating the know-how, which we now consider to be in a competitive advantage for Cellmedica to be able to develop these products. So that's a really good rationale for me as to why buy a company and generate the know-how. New technologies, suicide genes, multi-CARs, gene editing, things like that. We're still some way away from understanding their use. And certainly part of our decision is speaking with experts about these things. And in fact, in order to be here, I'm actually missing a fantastic event that we're organizing in London. We called a lot of the CAR-T and TCR experts at Cellmedica to sit them down to talk about all these things. So obviously I chose this over that, so obviously that's very important. But that's really how we're learning the space, how we understand. We're asking the people who are developing all of these things, you know, why would you use that? How would you use that? You know, based on what you see, is a suicide gene more valuable than $10 worth of steroids, for example, in a patient? And start to understand all of these technologies. And that takes us a lot of time. But we feel that by doing that, we're actually much wiser and have a much better understanding of the steps that are ahead of us and start to make some good informed decisions and choices.
Great. I know there was a question. You have a question? ahead.
I wanted the prospect came up, which then makes me think of cancer vaccines as defined 10 years ago. I mean, Prospect's been in clinical development for a long time. We've been told that there are quite a few phase 3 very old cancer vaccines that are making their way forward. What kinds of opportunities are there for the old cancer vaccines in this new environment? And should we be calling them cancer vaccines?
Yeah. Well, I think that I agree with Niels. I think that we're seeing this as kind of a burgeoning space. I think we're still interested. I will say it's a high bar. Obviously, there's been more failures than not. So we're waiting to see actually the readout of the Prospect data. Actually, it's that trial continues to enroll, and we see what's happening, or rather, we're waiting to see kind of events in that, and we should get that data. We're hoping this coming year to see if that pans out. That being said, I think that we're also looking to try to combine that as well and see if that's a good approach. But I think we are looking at it. I think it is a high bar to think about some of the old technologies, honestly, but not to say that rethinking and using some of the insights and findings that have come out within the immune space wouldn't be something worth talking about. But I think that clearly everybody's interested in, hey, looking at these colder tumors, how you can inflame them, how to create that response is one way to think about it. The only other thing I would just mention in this whole piece, which is I think the more novel your modality, the more rigorous and robust your CMC and manufacturing platform is going to have to be from a big pharma company. So oftentimes we're talking about preclinical data and clinical data, but that can often be a huge swing factor for us because, of course, that's probably by nature not going to be expertise for us. We're going to you to get it. So I think thinking through those investments often can swing the deal. We did a deal with Unicure, not in the oncology space, but in the cardiovascular space, partly because they really had incredibly robust manufacturing capability in that space. So that's an important consideration as you're thinking about novel modalities from our perspective.
So, well, one question. So before I get to that question, I just want to follow up with Baron, just because since you raised the question of vaccines, clearly, there's the concept of exogenously supplying a vaccine, maybe to a predetermined antigen. Historically, there's been more the Gamish approach, just grind up allogeneic cell lines or patient tumors and deliver them. And then I think it was Niels who talked about the kind of the danger signals, et cetera, which you either get from oncolytic viruses like the Cyoxis, or you can get from radiotherapy. So I'm just curious when you're out there when you're internally thinking about kind of your business development strategy, Baron, as well as when you're kind of presenting your story to your prospective partners, what type of traction or not are you getting? Do people raise the issue of, well, if I'm going to have immunogenic cell death, I'd rather have a vaccine? That's a more directed approach. So,
not an easy question, actually, to answer. I think, first of all, radiotherapy combination with immuno-oncology is nothing new. There are currently 90 industry-sponsored trials ongoing, so there must be some believers in this technology, otherwise nobody would spend the money on this. So essentially, from a business development strategy, of course, we are trying to discuss with those believers, other than to persuade non-believers in radiotherapy, and there are a lot of non-believers in radiotherapy, in particular in pharma company, because radiotherapy traditionally is not a sweet spot of pharma companies. It's a device business, perceived as a device business. There are no drugs for radiotherapy, radio protectors, radiosensitizers all failed for numerous reasons. So it's not really something when you go open a door to Paul's office and say, hey, Paul, I'm here with radiotherapy. He's not going to cry, hey, I waited for you for many years, right? And this is, so we have to distinguish, first of all, we have to find those people who really believe. And I think looking into those companies running combination trials with radiotherapy is identifying those believers. Secondly, I strongly believe that with our technology, we can increase the immunogenic cell death, and we have all the necessary data to show this. And so our strategy clearly is focusing on, first of all, the big players, because those are the established therapies which are available, where second products are coming up, where competitors are coming up. So the question is, how can I differentiate, let's say in the PD-1 business, how can I differentiate myself being third or second to market? And this could be an opportunity, for example, for us in saying, okay, you combine your product with radiotherapy, and you can make it patentable by utilizing our product. So this is for us actually a tool that we may provide to pharma companies to differentiate themselves from existing competitors, from second line, third line comers in this, I'm talking about targets now, to come very soon. I think this is the strategy to go for those products which are already matured in clinical development or already on the market. Alternatively, if we talk about those targets which are really relatively early in preclinical or even clinical development, I think we are much more open-minded to this, to make a more open deal, because in this kind of setting, we do not know who is making the race, right? In some areas, we know we have to, Pfizer is number one, so we have to talk to number 2 and 3, but in other areas, there are so many people in this field so that we are pursuing an open deal road at the moment. So it's not really an easy question to answer. Particularly, we haven't signed a deal, but are hopeful to do so.
Okay, thanks. So, sorry, you had a question, sir.
Thank you. I'd like to know whether there's any clinical data or any data whatsoever that shows whether the dual approach increases the side effects. If you have an immunotherapy and, say, radiation treatment, it might be one thing. If you have with another combination, it might be another. But is there any data showing that having 2 treatments might be more risky than one? Or for that matter, why not have many different treatments? Admittedly, there's a cost factor, but what's the limit on all this?
Good question. It sets up a number of other points I wanted to address. Well, we're going to go to someone who knows a lot about combinations. There you go, Paul.
Well, I think, and this is what I was trying to say earlier, I think in our earlier experience with both Yervoy and Opdivo, which was a PD-1 and a CTLA-4, initially we did see that, there was added toxicity, but then what we did is, looked at, but we also saw a lot more efficacy, and then we figured out a way in which to optimize both the sequencing and the dosing of both. Now, those are obviously, 2 therapeutics that you can do that with, but in some cases, the same idea that you could optimize over time, so that basically we've gotten, which is, much, much higher efficacy than you could get in monotherapy with a monotherapy-like profile. So, I think that's a good example in my mind. All of us, this is kind of a key piece, right? Doesn't matter, oncology, out of oncology, combining 2 drugs, you always have to be thinking early on about how are you managing the tolerability profile, going forward.
Yeah, I'd be curious to hear from you, Nils, about particularly like the combination with PARP inhibitors.
Well, that's too early to tell, which is probably a sentence we're going to hear a lot in 48 hours from now. But for Rova-T combinations and PARP inhibitor combinations, that's just too early to tell, and there's no data I could talk about. But in general, I would be skeptical that you can get the profound, meaningful efficacy from an immunostimulatory drug without any side effects. And even if you go back in history and go all the way back to the first half of the 1990s and vaccination experiments back then, if there were any correlations between clinical outcomes and other observed effects, those were with reddening, local induration, pain at the injection site, and things like that. So I'd say it's probably unlikely that you'll find an inverse correlation between clinical outcomes and side effects for IO drugs. So if we keep adding on doublet and triplet therapies, we should probably expect to see more of that, but it all goes back to how can we successfully manage that by having pulsatile therapy, metronomic dosing, adjusting the dose for each individual administration. And I think BMS has done a great job at showing us how we as a field could think about that. And the last thing you want to have are overlapping spikes in toxicity. Those need to be avoided. In the end, if we get to a point where you, in metastatic patients, if you just need to fry a single metastasis with radiation locally and you get abscopal effects affecting the other 10, 20, 30 metastases in different tissues and organs without any side effects, would that be an ideal scenario? Yes, absolutely, but we're a long way from that, I think.
Well, I'll just add too, it's also, I mean, I don't see there's necessarily a reason to set a higher bar for IO combinations than that's always been the challenge in oncology, because it's pretty much always been about combinations and trying to optimize. Now, the issue is that historically, and even still to this day, there's no kind of industrialized way to do that. Not in the model systems, right? I mean, the original development of taxanes and platinums, I mean, you know, certain kind of standard of practice, management practice, the realities of practicalities of actual drug development and treatment led to certain dosages and combinations being used, but there's no methodical, systematic examination of dialing up and down the doses, dialing in and out the sequencing relationship. But certainly in IO, that's incredibly important, the sequencing, because you can use combinations of checkpoints and co-stims, give them together, and you don't see any. In fact, it may even be antagonistic, whereas, you know, opening up a window between delivery of those agents can actually really improve the outcome, at least in the model systems.
I mean, the other thing I would say, what's interesting about versus the, you know, chemotherapeutics, where, you know, you're getting a lot of toxicity, right? And you're not sure whether that toxicity is actually indicating any kind of efficacy. Typically in the IO world, right, which is if you're, you know, if you're turning the immune system on, you know, and you might be turning on a bit too much, obviously, when you're getting these side effects, but at least it's telling you that the drug's probably active. So there is some initial benefit to seeing some, you know, therapeutic index associated with the IO drugs, and then trying to figure out, as we did with, you know, with the combination with Ipi and Nivo, which is a bit about the ratio between kind of turning on one aspect of T cells and then turning down another aspect of T cells to try to get kind of an optimal balance. And I think as we, you know, learn more about these different approaches, you know, we could be able to, you know, thread the needle on these. Even going to triple and, you know, perhaps quadruple therapy, because if you're getting up to, you know, 80, 90% response rates, that then is leading to long, durable responses, you know, and we've seen this in a lot of diseases. We've seen it in pediatric, you know, leukemias. So, I mean, I think you've seen it in HCV. So, you know, I think we'll figure out a way to get there.
Yeah, just one thing I wanted to add is the other thing we have to continue to learn and understand better is when do you look, how, and where. Because there's protocols out there where, from an immunological point of view, you could have questions that the point in time that was picked will actually tell you whether or not you induced an immune response in the right place. So I think that's another area where we have to continue to add on to our existing experience.
One would argue that, to your question about vaccines, that regardless of whether they were suboptimal or didn't have things like checkpoints to harness onto them, some of those trials, certainly those were not inactive, and that's not the reason they failed. They failed because the trials weren't carried on long enough, there wasn't enough revaccination to see that tail end separation, because if it's a marginal efficacy, as many vaccines were, you would really need to kind of wait to see, and that just wasn't logistically feasible or even known about at the time when, because most of those vaccines that are in development now started way back 15, 20 years ago at the NCI, right? So, any comments on this kind of therapeutic index question, particularly for...
Well, it's obviously very relevant to cell therapies, because as you increase the functionality of the cells, you could call that a combination. So getting a T cell to secrete IL-12 on top of, you know, whatever else it's meant to be doing is quite a big step. And obviously IL-12 can be very toxic. So yes, there is increases in the side effects. But as with everything else, it's a matter of being able to control that, or understanding how to control that. So in the early CAR-T trials, and obviously we're seeing that every time a new development comes into play, some deaths in clinical trials occur, which are obviously very unfortunate. But in a way, it shows us on the one hand where's the edge of the envelope and where we can push things. But at the same time, clinicians are now so much better at treating the cytokine release syndrome with steroids and tocilizumab, which they weren't 5 or 6 years ago. And whereas it's considered to be quite routine. So basically, as they are now, they are monitoring the patient. And of course, it's expensive because the patient has to remain in intensive care as they're experiencing this. But they're monitoring the patient and sailing as close to the wind as possible without intervening, because the overlying assumption is that there is a therapeutic effect that is going on there, and what we're seeing there is the tail end of that. And then as soon as things get out of hand, then they can intervene. The same goes with suicide genes or with on switches, whereas everyone is working now towards modulating them rather than having them as a binary effect. And so while there will be some side effects, there will be potentially a closing therapeutic window. At least we will have the control to remain within that therapeutic window and establish some therapeutic efficacy even in the presence of side effects. There's a lot to learn.
Yeah. The other thing I'll add, which is I think we learned a lot from the launch of Yervoy, which is ipilimumab when it first came out. And actually the way that we launched it was actually to focus exquisitely on safety, because one great thing about oncologists, they're very comfortable and used to seeing lots of toxicity, just given the older agents. But what we had to kind of reteach them was immunology, which they weren't so used to, and weren't used to seeing immune-related AEs. So where they would see diarrhea as kind of a very transient, very, kind of passive type of AE that you could just give Imodium for. No, for us, that was a serious side effect. If you saw that, you needed to get on top of that, because that could be indicative of a much larger kind of immune response in a negative way. So, I think that's the other piece, which is really thinking about ways in which you can, as you said about, cytokine release, that, pre-medicating, those type of things, where you can actually get out there from a medical perspective and help physicians actually understand how to manage some of these toxicities. But in general, I think the tolerability profile of immune therapies is really quite amazing, particularly as compared to, chemotherapy in particular.
Yeah, I have a question for Stefanos. You mentioned suicide switches, on-off switches. You could add to that modifications you could make to co-stimulatory domains and expression of multiple gene sets, and the sky's the limit in a way, right? So which variables do you prioritize for getting out of that CD19 B-cell malignancy space where we all know it's going to work, and make a very profound contribution to patient lives? But how do you prioritize these variables if you wanted as Cellmedica to go into solid tumors?
Well, that's a difficult question. So anybody who knows the answer to that, I will give you money. Actually, I will. But no, I mean, obviously, we look at things, like I mentioned before, it's a functionality and the specificity effect. So we consider, for example, the optimization of the scFv, which is what drives the specificity of the T cell, to give us the specificity that sort of leads to on-target effects, but not on-target off-tumor effects in the same way. Obviously, that's building on some published work, and we need to build onto that. The suicide switches, we call it synthetic biology. That's how people sort of like to refer to this. In some cases, it's much simpler than synthetic biology. It's sort of chemically induced dimerization. Like you take a molecule, you split it in half, then you use a small molecule to pull it back together, and then it works. But being able to modulate that to have the right functionality when you want to have the right functionality, how active does a T cell need to be? Nobody knows. We only find out when we put it into the patient and we realize that it may have been a little bit too much this time. We'll have to work out a different process. And it's certainly not the dosing part. Reduced doses don't have a smaller chance of generating cytokine release syndrome. If you have the cells that are going to be reactive in your preparation, they're going to do that. Two ways, I think, to answer the question. The first one is with some science. So there are a few technologies that we realize that the only way to really understand their worth is to use them, with our collaborators, or build feasibility studies and actually test some hypotheses based on which we will understand, yes, we think this might work. Do you need to gene edit, for example, things out of the cells to avoid them getting rejected or to avoid GVHD? I mean, in our case, with the NKT cells, we do not need to remove the TCRs to make an off-the-shelf product because all NKT cells have exactly the same specificity for alpha-gal ceramide. It's a pathogen-specific molecule, so TCRs are invariant. So then we need to start thinking about, and we are thinking about, do we need gene editing in order to cut out HLA to prevent them from being rejected? Can we use shRNA? Can we use another technology along those lines that might have the same effect? There's only one way to answer this, and go back to the lab, do the experiment, come back and say, that's what we need. And obviously, this is a partnering panel. So, what implications does that have on partnering? Because obviously the concern you always have is you go to a partner, you build a feasibility study, you validate their technology, and then you have to negotiate the terms with them once you've actually done all the work to validate and de-risk the technology. So, and as these are things that you have to consider while you're doing that. Some of this you can do yourself, some of it you do with a partner, and sometimes you have to pre-negotiate some of the terms. But certainly, so the one answer is do the work and come back with an answer. The other way to think about it is if you're talking about a new target, speak with someone who's worked with that target, a new antibody, a new SCFE, and that's certainly something that we are very actively searching here. Is targets to build into these products that we have, either TCRs for the UCL collaboration or SCFEs that we can develop in Zurich and send to Baylor. People who've worked on the targets, they understand the histology, they understand the patients that will be expressing these targets. In many cases, they have treated these targets with monoclonal antibodies. So we can learn a lot from these people. And that's what I think is going to be one meaningful interaction we can have with pharma companies who have development, discovery capabilities around different targets, that we can get our hands on targets which we can believe in. And then it's the overlap of the expertise between two different entities, two different organizations that come to a better answer. Again, some validation either in clinical trials or if possible in preclinical models is almost certainly going to be required.
So let's bring this back to the beginning, and since we have 7 minutes something left. So this idea of combinations, and you raised that it is all about the partnering. So, and this gentleman raised the point indirectly about the combination therapies and the price. Well, let's not talk about price, let's talk about value. And we'll have a session on value and combinations and the proliferation of people still developing checkpoint inhibitors at our Cancer Progress meeting in March. Please come, there's my plug. But it raises the point of, if you're a small biotech, if you have a novel agent and you're going to need to combine it with a checkpoint, at some point, you already have to be asking, and our clients that are small biotechs are already asking, all right, well, what is the benchmark, not just for approval, but what's going to be the benchmark from kind of the market access side of this equation as they're getting increasingly powerful? Or they're asking, should we access a company, license or acquire, as Aduro did with Bionovian or others have done, to control our own checkpoints? We'll have more pricing flexibility by having our novel platform or agent combined with our own checkpoint inhibitor. So I'm curious to hear, certainly from Paul and from Niels, kind of how they're thinking through this issue of the challenges of combinations, not from the clinical scientific, we've touched on that, but more from kind of the patient management and the value aspects that this brings certainly Europe's already there, but the clouds are gathering more quickly even in the US. That's my very subtle allusion to the election, but that's all right.
Well, I think always our approach has been, to really look for these transformational type, impacts. I mean, I think when we initially saw some of the early lung data, for example, with PD-1, and then saw the combination of it, I think it gave us a lot of conviction around, hey, that kind of response where, or what we've seen in melanoma, 10 years where people, of those who have responded are, still alive from what would have been, absolutely deadly disease in just a couple months in the history, I think gives you a lot to go backwards and really argue the value for that. I would also say, as I said earlier, that there are a lot of precedents for how to kind of work out value sharing, across companies, I think. But in the face of a very, efficacious, very large therapeutic index. So, I mean, I think as smaller companies are thinking about, hey, where do I go? you're going to, and I get it, it's risky, but that's where you need to go. You need to go where there's going to be a clear unmet, need that we're not about to get to, right, between Nils or myself or our competitors. So, that takes some risk, but we're all taking some risk. So I think that would be my advice, is to really focus on, hey, where do we think we could have really a transformational approach? And, there are a lot of tumors out there, and there's a lot of tumors that we're not going to get to, or there, are places where, we're not yet, there are people still progressing, there's still people not responding. That there is a lot of unmet need there. And I think, at least for now, I think in oncology, there really is a willingness to pay for value.
Yeah, the only thing I can add to that is maybe 3 points. One, I totally agree with Paul that we have to shoot for clinically meaningful differentiation with novel combinations. With just architectural minor incremental improvements, there's going to be little gain. Secondly, I think companies, research groups, large and small companies should look long and hard at the types of experiments and combinations they want to test, because there's going to be many that are going to be purely additive. But to ferret out the ones that will be truly synergistic, that is going to be the challenge. But with just additive efficacy and activity, it is less likely that you will have a differentiated value proposition. So I think that is quite important. Lastly, I think I strongly believe that the markets will continue to pay for innovation, but that goes full circle back to it has to be clinically meaningful.
Yeah, so Baron, I want to bring you back in for the discussion here on this challenging topic. But
No, I would totally agree, but my answer would be different. I would say come to us, right? Because I think the alternative approach is not just to combine, but to as I said in the beginning, it's to prime the immune system to work better, right? And this is the alternative approach to, this needs to be in a meaningful setting, this has to be good output, everything, but in the end it comes back to the biology. How can I fine-tune the immune system to respond better, to work better in favor of killing the tumor? And this would be our proposition, to say, don't only think about combination, also think about the enabling technologies which are where the clinicians are surrounded with, right, like radiotherapy. Try to think out of the box, and I think our approach is just one approach which is available, and I truly believe it will work out, and that we will be seeing more radiotherapy combinations over the next couple of 24 months or so. But I think it's not only about combining these different existing products, it's also to take new avenues, to think out of the box, because this clearly opens also not only for the patient's perspectives, but also for the companies in terms of commercial attractiveness.
Yeah, I just want to echo, because I think we're getting close to the end, what both Paul and Niels had said in terms of, if you're a small biotech, don't just focus on kind of doing a proof of concept in, kind of a model of melanoma that's going to put you in a situation where the readout is going to say, well, we're going to have to either improve on a Ipi-Nivo combination, or we're going to have to, increase that by such a, it's just not going to be feasible. So kind of doing that proof of concept in a model system that's going to be suggestive of something that's going to be clinically relevant, or what we call kind of proof of relevance, I think is the key, right? So if you're going for primary refractory or progressing patients in melanoma as your concept, do the models that are going to enable some conclusions to be drawn about the potential applicability in that, or go after the more intractable, colder, or less immunoresponsive tumor types, so that you're actually doing your early model work in a setting that is going to be clinically interesting to someone, not just showing that it technically works.
I could add to that, and the proof of concept and the quality of the proof of concept is actually very important. So as has been said by others, and I think as a previous panel that I was hearing the same story, is that it's very difficult to find meaningful animal models to understand how these things work. So that actually doesn't mean that we get to skip the whole science altogether and just put it in patients and see whether they get better. But it actually raises the bar in terms of the quality of the data that we have to generate out of our clinical trials. So the immunomonitoring and all the other additional types of information that will actually give us some more comfort, not just on a proof of concept, but also on the mode of action of this therapeutic. Because eventually, if we're ever going to be able to analyze and explain the clinical data, we need to have some understanding of what's the mode of action, and what cells are being activated, what cells are still not being activated. Do we still pick regulatory T cells in the circulation, in biopsies, and really do the extra work to understand, and so when it's time to actually take it out, either for partnering or for regulatory discussions for an approval, we actually can explain what it is that we have in our hands.
Great, good point. Thank you, Stefanos. One last question, if anyone has one. Otherwise, you're off to cocktails. No? All right, great. Appreciate everyone's attention. Thank you, and thank you for the panelists.